He also takes advantage of the people's vulnerability.
And then 15,000 of the people are coming to live in Africa just because they are building a road for you. We don't have the knowledge to maintain that infrastructure. And now they're just gaining citizenship because they're here for a long time. If you live here for 15 years building a road, of course you can get citizenship. And now there's a land crisis in Africa. Where will Africans live? I am sorry for my continent.
And we will continue with our experiments and investigating photosynthesis. Today I would like us to concentrate on light and carbon dioxide. And now we are going to start with a few new investigations. The 4 investigations that will be important to us will be: is light needed for photosynthesis? Is carbon dioxide needed for photosynthesis? Is it necessary that chlorophyll must be present? Is it needed? And then, is it oxygen that is produced during photosynthesis? Today, we are only going to concentrate on 2 experiments, and that is light important for photosynthesis, and is carbon dioxide needed for photosynthesis? Just to help you to remember, where is light important? We remember that photosynthesis takes place into 2 phases. Yes, we have phase number 1. Phase number 1 is the light-dependent phase, a phase that cannot take place if light is not present. And in today's investigation, we are going to see, is it really the matter? We call it photolysis because this light will split the water molecules into the hydrogens and the oxygens. Now, and then we are also going to look at the second phase where carbon dioxide is reduced to form carbohydrates. That is the light independent phase. This phase does not need light. But as we said yesterday, if the light phase did not take place and there are no hydrogens coming into this phase to have hydrogens, carbons and oxygens that will form the carbohydrates, this phase will also not take place. Now, and that is what we are going to study today. So, we said, for investigations to take place, we will need 2 groups. We can only take 2 plants, but that is a bit risky. The more examples that I have in an investigation, and I can work with an average, the more accurate will my results be. But I will have 2 plants. The one will be called the experiment, where I give everything that is needed, in this case for photosynthesis. So I will need water, I will need the correct temperature, I will need chlorophyll, I will need carbon dioxide, I will need light. If I am investigating light, then I will remove the light in this experiment. In the control, I will give everything, even the light. So, why? I want to compare the two investigations, and I want to prove that if the light is removed, no photosynthesis takes place. You agree? Good. So, we also said, I have to destarch before I start the experiment. Can you quickly tell yourself why? What is the answer? Is this your answer? A plant is left in the dark for 24 to 48 hours. That is destarching. That all the stored starch is used up. I want to prove that the starch that was formed during the experiment was due to the factor under investigation, or was lacking, starch was lacking due to the factor under investigation. And of course, before I start with the experiment, I do the starch test to prove there's no starch in this plant. Now I have my control and my experiment, and I now have something to prove. Okay, and now let's look at a setup experiment investigation about, is light really necessary for photosynthesis?
Now in the next practical that we can do, we want to, the aim of the practical is to see whether light is indeed necessary for photosynthesis. Now, what would we need? We would need a destarched potted plant, a plant that has been left in a dark cupboard for about 48 hours, and you will see that we have now placed foil over parts of the leaf, blocking out light under the foil. You can decide exactly what pattern you would like to make. What you would also need is all the apparatus that you have used previously to test for the presence of starch in a green leaf. The method of this practical would be, once you have destarched the plant and covered parts of those leaves, you would allow this pot plant in the sunlight for at least 4 to 8 hours. Once we have done that, I would remove a leaf from that pot plant and repeat the test for the presence of starch in the leaf. When doing this practical, it's important to emphasize what the result of this practical will be. After testing for the presence of starch, we will see the following. After doing the test for starch, the last step in the test of starch is when we place iodine solution onto the pale leaf. On this side, we will see that there was an immediate positive color change. Iodine solution changed to blue-black. But on this side, you can see that iodine solution stayed the brownish color. Now, when doing this practical, our aim was to see whether light is necessary. So this part of the leaf that was left uncovered would be the experiment, but the part of the leaf that was covered by foil would then serve as the control of this experiment. Once we have our results and we've observed that when light was allowed, we saw that a positive color change occurred, and when light was not allowed, the covered parts did not change color. We can now take those results and see that our conclusion would be light is definitely necessary for photosynthesis.
So, grade 10, we have a covered part and we have an uncovered part. And you have to now make a label diagram to show the results. And it is so important that you make sure when you do this drawing that your drawing looks exactly the same as the drawing that they gave you. Okay? So I will then make sure that that part that is covered is also the part that I have covered there. Now, and now I will have to add labels. Okay? And of course we do this with a ruler. And now this part was covered. So this part will be yellow-brown. And this part was blue-black. And this part was blue-black. And now, as the Tannie told us in the video, which part is the experiment? The part where the light was taken away. So, if we use a highlighter and we say the experiment will be this part. Yes? That part is the experiment. Because we said this part had all the requirements for photosynthesis except the factor that is under investigation. Good? And then we have the control, and this will be the control. Why do I say so? Because the control undergoes the same as the experiment, except that the independent variable is kept constant. Oh, so why are we talking about now about the independent variable? I just want to explain it to you like this. If light is there or not there, and I draw a graph, I will have light on the x-axis because the light can be changing from very low to very high because light is what I want to show as the factor. So in our previous slide we say except the factor that is under investigation or the independent variable. Light was in this case our independent variable because it was there or it was by my control taken away. And that is why we say it is the independent variable. I hope that you do understand that. Right. You have experiment number 2 on page 161. I want you now read through that experiment and see there are a few questions. Now, question 6 and 7. Answer that in your book. Make a heading, Experiment 2, page 161, the day after day. And it is about photosynthesis experiments. And answer that. Now, we are going to continue to look at carbon dioxide is necessary for photosynthesis. Okay.
During this practical, we are going to see whether carbon dioxide is necessary for photosynthesis. Now, what we need is 2 destarched pot plants, and to do that, we need to place these pot plants into a dark cupboard for 48 hours. Then test one of the leaves out of each of these pot plants to make sure that all the starch has indeed been used up and that there is no starch present in the pot plants at that stage. And then you place these pot plants into a bell jar. Use pot plants that are more or less the same size. In the control, we have soda lime in the funnel, and in the bell jar, sodium hydroxide in the beaker, and both these absorb all the carbon dioxide and make sure that no carbon dioxide actually enters the bell jar. On the other side, we have the experiment. In the funnel, we have normal chalk, and then inside the bell jar, it has water. The little beaker has water, but it could also contain calcium carbonate so that we do have carbon dioxide available so that this plant can indeed photosynthesize. Now we place these 2 bell jars into the sunlight for about 48 hours, and then you are ready to test for the presence of starch in both of these pot plants. The result of one of the leaves taken from the control, we will see that the leaf stays a yellow-brown color, which indicates no starch is present in the control. But in the experiment, we see that the leaf has turned to a blue-black color, indicating the presence of starch. Our conclusion will thus be that carbon dioxide is indeed necessary for photosynthesis, because we saw that starch is present when carbon dioxide is present.
Okay, so let's look at the experiment. Soda lime prevents entry of carbon dioxide. I have a bell jar, and normally we can even seal them by having Vaseline, petroleum jelly or Vaseline added here. Added here at the bottom, so to make sure that it is really sealed off. And we will do everything on both investigations. What do we see here? We see that they seal off even the soil. Why? Because remember, in the soil, there are a lot of microorganisms. And these microorganisms respire, and they will give off carbon dioxide. We want to make sure that there is no carbon dioxide in this jar. Good. I think that this is then the experiment, because our book says the experiment is where I have all the necessary factors for photosynthesis except the one being under investigation. Okay, so now I want you to concentrate on, why did I put in sodium hydroxide? Because all hydroxides absorbs carbon dioxide. So even if there was any carbon dioxide in this air, it would have been absorbed. Good. Soda lime absorbs it there as it moves. If there are any left in the air, absorb there, prevented from the microorganisms to release any carbon dioxide. On this side, we have the air coming in, and we know air contains carbon dioxide. Now, here in this picture, there's water, but I can add a carbonate, any type of carbonate in here, which will release some carbon dioxide to make sure that this one is filled with carbon dioxide. According to our book, the control is then this one. Why do I think this is the control? Because all the necessary factors, even the one that is being under investigation, is present here. Do you understand this? So here I added some more viewpoints and some more small important stuff to make sure this one has no carbon dioxide. All hydroxides absorb. Hydroxide absorb carbon dioxide. And in this one, there's water, but I can also add a carbonate solution there that will release some carbon dioxides. This one is the control because it contains all the necessary information. This one is the experiment because it has all the information except the one under investigation. Good. And that brings us to the end of this lesson. Answer the questions on experiment 2 and 3 on page 161. And please look, I also add a PDF with some good questions. On it, I want you to also try those questions to help you to practice and investigate photosynthesis.
One Africa TV's #LearnOn1 and Hollard Life are teaming up to give lucky teachers an awesome goodie hamper. All you have to do is tell us if you'll recommend #LearnOn1's free, quality, supplementary lessons to your students. Simply SMS a yes or a no, including your name, the town you live in, and the school you teach at. It's that easy. Invite learning in today with #LearnOn1 and Hollard Life. Competition closes on 16 October 2020. Standard T's and C's and SMS rates apply. For more information, visit learnon1.org.
When you watch football on GoTV, you know what you want. You want superstars.
What about that?
The biggest names in the game. And of course, the best leagues in the world. Yes, you want explosive football action. And this new football season, we're serving it to you on a shiny new platter with our revamped Super Sport channels. All you have to do is tune in and we'll make it worth it. So get and stay connected to GoTV for football worth watching. GoTV, live it, love it.
Get uncapped internet from as little as $500 million a month. Contact us now or find us on Facebook. Whitehall, your internet service provider.
This morning, the investigation on photosynthesis. And like we did in the previous lesson, we are going to first look at why. Today we are going to look at chlorophyll and is oxygen released during photosynthesis. So, again we are busy with, there are two phases. The first one is the light-dependent phase, during which the reaction is photolysis. And the second one is the light-independent phase, where the reduction takes place. Now, in our first experiment, we are going to see whether chlorophyll is important so that photosynthesis can take place. And in the second one, we are going to look at, is it really oxygen that is produced during photosynthesis? So both the investigations today will be part of the stage 1 light-dependent phase of photosynthesis. Good. The first experiment is then where we are going to look at, is chlorophyll necessary for photosynthesis?
In this practical, we will determine whether chlorophyll is indeed necessary for photosynthesis. For the practical, you would need a destarched leaf which is variegated, which means that there are parts that are green, which contain chlorophyll, and other parts, if you look on the margin here, you will see that no green pigment occurs in that leaf. And that's why it is colorless. Now, what do we need to do to destarch this pot plant? We would have to place it into a dark cupboard for about 48 hours to ensure that all the starch that has been produced previously will have been used up, even the parts that would maybe be in the non-colored parts of the leaf. Once we have left this pot plant outside for a few hours, I will then pick one of the leaves and I will test for the presence of starch as shown previously in the first practical demonstration. Now, once we have done that and we have tested for the presence of starch and we have added iodine solution to the leaf after the test for starch, we will see that the parts that did contain chlorophyll has now tested positive for starch. But on the margin, where we see that no chlorophyll was present, we will see that the iodine solution has stayed its brownish color. The middle of the leaf, if we compare it to this leaf, shows that the parts that contain chlorophyll have turned to a darker color. Now, I have placed the iodine solution onto this leaf, and the longer I leave it, it will actually turn even darker. So we can see a definite difference. The longer the iodine solution stays on that leaf, it will turn even darker to a blue-black color.
So we have seen a variegated leaf. Here on our premises,